on His Majesty's service. Olfactory receptors. Olfaction is the sense of smell, the ability to detect the smell of substances, odorants, dispersed in the air. In humans, olfaction is considered the least important of the senses. Nevertheless, odors provide information about food, plants, people, animals, and other aspects of the environment, and olfaction can influence feeding behavior and social interactions. Olfactory receptor cells are bipolar neurons. They are localized in the pseudostratified epithelium of the olfactory mucosa, which occupies an area of 5 to 10 square centimeters in the upper posterior part of the nasal cavity. Olfactory sensory neurons have short dendrites and long axons. Each dendrite forms a knob-like protrusion, dendritic bulb, which projects above the epithelial surface and carries 10 to 20 cilia. Olfactory cilia are considered to be non-motile. Olfactory axons extending from the basal part of the receptor cells form bundles of the unmyelinated olfactory nerve, which reaches the olfactory bulb of the brain. Olfactory sensory neurons are located between the supporting epitheliocytes. They are characterized by microvilli on the apical surface, numerous mitochondria, profiles of smooth and rough endoplasmic reticulum, and lipofuscin granules in the cytoplasm. These cells perform trophic and support functions. On the basement membrane, one can see small, rounded basal cells. They serve as a source of regeneration of olfactory and supporting cells, which have a short life cycle. Secretory protrusions of the olfactory or Bowman's glands are located under the basal lamina of the olfactory epithelium. Glandular cells possess lipofuscin granules in the cytoplasm. Together with lipofuscin granules of the supporting cells, they give the olfactory mucosa yellow color. Excretory ducts pass to the epithelial surface to discharge the proteinaceous secretions which form mucus. Odorant transduction begins with odorant molecules selectively binding to G protein-coupled receptors located in the plasma membrane of the cilia. Odorant-binding proteins that are secreted by supporting cells into mucus are believed to increase the solubility of hydrophobic odorants by binding them and delivering them across the mucus to receptors. After interaction with odorant receptor activates associated heterotrimeric G protein. Then alpha subunit of this protein activates olfactory-specific adenylyl cyclase, leading to increased intracellular levels of cyclic AMP and opening of cyclic nucleotide-gated cation channels. Subsequent entry of calcium and sodium ions into cilia leads to depolarization of a neuron. The rise in ciliary calcium ions concentration activates chlorine channels, which are also present in the ciliary membrane. The opening of these channels causes an efflux of chlorine from the cilia, which further depolarizes neurons. This produces a large amplification of the primary signal. Depolarization leads to action potential generation and its transmission to the olfactory bulb of the brain. In the continued presence of odorant, sensory neurons adapt. Adaptation is achieved by reduction of action potentials due to increased calcium ions binding to calmodulin and other calcium-binding proteins, which decreases the sensitivity of cation channels to cyclic AMP and activates the phosphodiesterase, which hydrolyzes cyclic AMP to AMP, and extrusion of calcium ions because of activation of sodium-calcium exchangers, which returns calcium ions concentration to basal level and reduces the depolarization. In many vertebrates, besides the main olfactory system, there is an accessory one, the vomeronasal system. The vomeronasal organ was discovered by Frederick Ruch in 1703. The vomeronasal organ is located in the soft tissue of the nasal septum above the hard palate. It consists of a blind sac with a duct opening anteriorly. Its mucous membrane is similar to the olfactory mucosa. The sensory neurons of the vomeronasal organ respond to volatile pheromones, which are chemical signals that regulate a series of innate behaviors, such as reproduction and aggression. The vomeronasal neurons express two families of receptors, the V1 and V2, which are G-protein-coupled receptors. The perception of pheromones is important for the formation of sexual behavior and emotions. For a long time it was believed that in humans the vomeronasal organ is reduced and has no functional significance. Nevertheless, some studies with the use of direct and endoscopic observations, classical histological stains, and electron microscopy, computed tomography, and magnetic resonance imaging showed that at least 1/3 of people have it active. In 2001, a second family of G-protein-coupled receptors, known as trace amine associate receptors, were described in the olfactory epithelium. The term refers to tryptamine, octopamine, and beta-phenylethylamine, which are derived from aromatic amino acids and are present in mammalian tissues at much lower concentrations than the classic biogenic amines. 6 trace amine associate receptors were identified in humans. They are believed to detect pheromones in rodents. Maybe in humans they also play the same role, although its function in humans is widely debated upon. Olfactory receptors are not exclusively expressed in the olfactory sensory neurons. They are also observed in many human tissues, including the brain, adrenal and thyroid glands, and other organs which are named in this slide. For most of the ectopically expressed olfactory receptors, limited data are available for their function roles. They have been shown to be involved in the modulation of cell-cell recognition, migration, proliferation, the apoptotic cycle, exocytosis, and pathfinding processes. Thank you for attention.